This video is for the answers for the worksheet for chapter 5, which has to do with the purification and characterization techniques of proteins. Part A is a match of the terms that we have on the column on the left with the definitions that are on the column on the right. Let's get started. So, the BCA assay, the answer is G. As you can see, that is a protein quantitation technique where the amount of copper 1 is related to the amount of protein that is present in the sample. The answer for 2 is E, which is a recombinant vector is what's formed when you are linking or ligating a PCR product and a vector. 3, which is NMR spectroscopy, is letter F, which is an analytical technique that is utilized to identify proteins based on the distances between the hydrogen atoms. Number 4, page, is letter B, specifically because it says a technique that separates denatured proteins. We're talking about SDS page. Remember that native page does not have samples that are denatured. And this technique is separating the samples according to mass. Number 5 is H. The PCR product is going to be the DNA of interest. 6 is C. UV spectroscopy is a technique to measure the quantity of proteins, specifically by looking at the absorbance of aromatic amino acid residues. Number 7 is letter A, which is the vehicle for protein expression. And lastly, number 8 is D, which cell lysis is another term for referring to breaking the cell open. Let's move on to part B. On part B, as you can see, this is the new graphics to talk about what are going to be the sediments that are going to be present once we use the technique of differential centrifugation. For this, you can relate it to slide 6 on the chapter 5 presentation. So, after the first centrifugation at 800 G, understand that the first sediment is going to be the nuclei. Nuclei is the plural word for nucleus. Then when you take the supernatant, as you can see in the words, and then you spin it at 15,000 G, the next sediment that is created has 3 different components: mitochondria, lysosomes, and peroxisomes. When that's supernatant, then it is spawned by utilizing 100,000 G. Then the next sediment that is forming has the plasma membrane, and the components of the ER. Then after you take the supernatant there and you spin it at 200,000 G, understand that at that point you have in the sediment the ribosomes, and the supernatant, the last supernatant, is going to be the soluble cytosol. And in that soluble cytosol will be where your protein of interest that was expressed is found, unless it is insoluble, so then it will be found in the sediment. Let's move on to the next part, part C. On part C, we are going to be dealing with the technique of size exclusion chromatography. In size exclusion chromatography, as it was explained in lecture, the first thing that is going to come out is the protein that is the heaviest. Okay, remember that the unit that is utilized here is kilodaltons. So the ordering of evolution for the proteins that were given in this instance is hemoglobin, followed by triose, phosphate, isomerase, followed by myoglobin, and the last one is lysosome. Or sorry, the lysozyme. Moving on to Part D. Part D reads, determine the product or products for treatment of the following peptides with trypsin, chymotrypsin, and cyanogen bromide. Assume you have 3 samples and they are treated separately with each of these enzymes. Okay, so when it comes to this, understand that we need to first define what are going to be the types of lyses, or in other words, how are these enzymes going to be cutting the particular peptide. So understand that when it comes to trypsin, it cuts at the end of 2 specific residues, arginine and lysine. Chymotrypsin, on the other hand, is going to be cutting at the end of any of the amino acids that contain aromatic residues. So it could be tyrosine, tryptophan, and phenylalanines. And lastly, when we have cyanogen bromide, understand that this is going to be cutting at the end of methionine, and understand that this has a specific feature because methionine during this lysis, okay, or this cutting of the enzyme, this hydrolysis, is going to turn into a brand new compound that is called a lactone. Okay, so now let's create a line to divide these so I can illustrate the different peptides that are going to be created. Okay, so when it comes to trypsin specifically, cutting the peptide that is given in bold, I'm going to use a highlighter to illustrate where the enzyme is going to be cutting. Okay, so as you can see in this given, this enzyme is going to cut right here in between lysine and asparagine, and is also going to be cutting in between arginine and methionine. So those 2 are going to be the sites. So in this case, you're going to be forming 1, 2, 3 peptides. Okay, so the first peptide is going to be methionine, tyrosine, alanine, serine, phenylalanine, methionine, valine, lysine. The next peptide is going to be asparagine, histidine, tyrosine, cysteine, glycine, arginine, and the last peptide is going to be methionine and tryptophan. So that's the lysis specifically for trypsin products. So I'm going to erase my work and introduce the ones for chymotrypsin. Okay, so I'm going to use a different highlighter. So chymotrypsin, I'm going to illustrate it by using this purple highlighter. And as you can see, it's going to cut at the end of any amino acid that contains a side chain that has an aromatic side. So as you can see, every time we find phenylalanine, tryptophan, or tyrosine, that's where we're going to cut. So as you can see, when it comes to this, is going to cut in between tyrosine and alanine, between phenylalanine and methionine. Then as we move right along, it's going to cut again after the tyrosine and the cysteine. And then after that, there's no more cutting, because since our last amino acid is tryptophan, then there's nothing to cut. So as you can see, in this product, we have 1, 2, 3, 4 peptides formed. The first one has methionine and tyrosine. The second one has alanine, serine, phenylalanine. The third one has methionine, valine, lysine, asparagine, histidine, and tyrosine. And then the last one has cysteine, glycine, arginine, methionine, and tryptophan. The last work that I'm going to illustrate is going to be the one for cyanogen bromide. Okay, remember the cyanogen bromide is going to be cutting after each methionine. Okay, and that methionine is going to be turning into a lactone. Okay, so these are going to be the products if we take the peptide and we cut it with cyanogen bromide. We are going to create a cut between the first methionine and that second tyrosine. Then we are going to do a cut in between the second methionine and that valine. Then we are going to be cutting again in between that methionine and that tryptophan. Okay, so let's make notes here. Remember that whenever we have a cut with cyanogen bromide, we're going to be changing this methionine into a lactone. So the second peptide is going to look like, so the first one is not a peptide, it's just going to be a lactone. Okay, the second one, it is a peptide. So this is the second fragment. It is your first peptide because there are links between amino acids. But remember that your methionine is going to turn into a lactone. So that means that the first peptide that is produced in this segment is going to be tyrosine, alanine, serine, phenylalanine, and then the last residue is a lactone. A lactone is not an amino acid. Then when we have the next peptide, this is the next segment. So the second one that is formed is going to be valine, lysine, asparagine, histidine, tyrosine, cysteine, glycine, arginine. And remember that whenever we have that methionine, that's going to turn into a lactone. And lastly, the reason why I don't count that last tryptophan, okay, is because once we form that last lactone, we're going to have a single amino acid. You need at least 2 amino acids linked to one another in order to have a peptide. So there's only 2 peptides formed when we treat the highlighted peptide given with cyanogen bromide. Let's go through part E. The problem reads, in your laboratory, you plan to use ion exchange chromatography to separate the peptide shown. Let me just fix that. Below from a mixture of different peptides at pH 7.0. Should you choose a matrix containing DEAE, meaning an anion exchange, or CM, which is a cation exchange matrix. And then you have to explain your reasoning in 3 to 5 sentences. Now, the first thing that you have to take into account is that the peptide that is given, you need to think about what is going to happen to it when I have it at pH 7.0, okay? So the first thing that you have to take into account here is how are my amino acids going to be affected at this pH? So to solve this problem, understand that you need table 3.2 that is given in the slides and utilize the values for the pKa's for the amino sites, the carboxy sites of each of the amino acids, in addition to the ones that are given for those side chains that are affected by pH, okay? So if I rewrite the enzyme given, okay, understand that in the first amino acid, which is glycine, it's nitrogen. So the nitrogen that I wrote, that corresponds to the amino nitrogen next to the glycine, is going to be exposed. Then I'm going to write all the other residues that are present. So as you can see, I have D, which is aspartic acid, valine, and then I have glutamine, proline, serine, leucine, another glycine, lysine, and then I have glutamic acid, I have serine, arginine, alanine, 2 more lysines, a phenylalanine, another glutamine, then I have arginine, and I'm just writing it down just for space. And lastly, I have another glutamine, and the carboxy terminus, meaning the carboxylic acid of that last residue, is going to be exposed. So remember, this is the carboxy terminus. Now, if I look at the values for the pKa's that I have in table 3.2 and compare it to the pH at which I'm treating this enzyme, then remember, anytime you have a value of pKa that is lower than the pH given, we're going to be losing a particular proton, okay? So now I'm going to highlight the residues that are affected, okay, by the changes in pH, okay? So the amino acids that are affected by the change of pH, okay, because their side chains could be deprotonated or they could retain the proton, is going to be that aspartic acid, lysine, glutamic acid. Of course, I already highlighted lysine, so I'm just highlighting it again. My arginines. And that's pretty much it. Because what happens is that the side chains of the ones that I highlighted in green are going to be affected by the changes in pH, okay? In addition to that, of course, my amino terminus in the first amino acid and the carboxy terminus in the last amino acid will also be affected, okay? So let's get started. By looking at the values of the pKa's, you will see that the amino terminus in the glycine is going to be remaining protonated, okay? Looking at the pKa of the side chain in that aspartic acid, you will know that that remains negative. Lysine, because of the pKa of the side chain, is going to be positive. That glutamic acid is going to be negative. Arginine is going to be positive. Again, we already have those lysines being positive. Arginine is going to be positive. And when it comes to the carboxy terminus, is going to be negatively charged. So the first thing that you have to do in this problem is determine the overall charge, okay, of this peptide. And again, so if you add them, the first two charges equal 0, the next two charges equal 0, then we have 3 positives, and the last two charges equal 0, so the overall charge is positive 3, okay? When it comes to deciding if you're going to utilize a matrix for anion exchange or cation exchange, remember that you have to understand what is anion exchange and what is cation exchange. When it comes to cation exchange, the matrix is going to be negatively charged. So Cm is negatively charged. When it comes to DEAE, which that is just an amine compound that is bound to a bead, understand that it is positively charged. And since your peptide overall is positively charged, then you must use a Cm, meaning a cation exchange matrix. And again, the instruction says that you have to do all of this in words, in 3 to 5 sentences. So the process that I illustrated, you need to convey it in sentences. Let's move on to the last part of the worksheet. Part F, as you can see, you're given some information in a table. And in this table, column 2 gives you the isoelectric points, which is going to be utilized on part B. And the third column, it gives you the molecular weights for the following proteins that are in that sample. So if we have a closer look at what the question is asking, as you can see, you're going to utilize the information of the table. We're basically going to be utilizing on part A. Okay, we're going to give the order of migration from top to the bottom of the gel in SDS page. As we discussed previously in the definitions that we illustrated on part A, we are going to be arranging these molecules by molecular weight, in other words, size. So understand that as we have to arrange them in size. The high molecular weights are going to be on the top, and then the low molecular weights are going to be in the bottom. Understand that to give you guys a guideline why that is, on lane 2, I said that we have a ladder. And a ladder, it is utilized to illustrate how these proteins run always when doing this type of experiment. So understand that these proteins always run at that molecular weight. So now, if I give these proteins in the sample a letter, okay? So let's say that I put a letter next to aldolase, so I'm just going to label this as A, B, C, D, E, and F, then that is just going to be a different way that I can label it on the gel, okay? Understand that for the exam, I may not give you the ladder. You need to understand how things run in an SDS page, okay? But to give you guys the illustration, as you can see, we have the units in Daltons. Previously, we learned that Daltons can also have kilodaltons, okay? So if we do the conversion in 1 kilodalton, there's 1,000 daltons, okay? So based on that, remember that we can tell that the aldolase is 40 daltons, The cytochrome C is 14 kilodaltons. That lactase dehydrogenase is 18 kilodaltons. Ornithine decarboxylase is 75 kilodaltons. Phosphoenolpyruvate carboxylate is 74 kilodaltons. And lastly, pyruvate kinase is 62 kilodaltons. Okay, so if we give the order of how they will look, I will draw them in the gel, but again, you just need to be able to write them down. If I illustrate it on the gel, we're going to have around 80 letter D, followed by letter E, followed by F. As we move right along, around 40 kilodaltons, we will have letter A. Then in the bottom of the gel, we'll have C, followed by B. And that is the order in which they will run in SDS page. Now, the last problem that we have on the worksheet talks about what is going to be the distribution in the pH gradient, okay, if you use an isoelectric focusing gel. The main thing that you have to remember is when it comes here, since you're assuming that you are running this from anode to cathode, okay, understand that in the slides it was explained how in isoelectric focusing gel, the anode is going to correspond to the low pH, okay, cathode is going to be high pH. So that means that the pH is going to be increasing as it's moving from anode to cathode, okay? So since that is going to be the trend, then you have to accommodate these in increasing pH units, okay? Remember that the isoelectric point gives you what is going to be the value in which we have a pH that makes your overall protein be zero. And in isoelectric focusing, the protein is going to migrate towards the pH that makes it have an overall charge of zero. So that being said, if we accommodate the sample that we have and we utilize the information of column 2, the order will be D, followed by E, followed by F, followed by C, followed by B, followed by A.